Good morning, good afternoon, people. I mean, depending on where you are, it's this is a great event, and I'm proud to have been invited to speak. And it's great to see so many old friends in the audience. What I'm going to do is briefly summarise the work of a consortium that has been funded by the WHO for some years now, with the simple goal of trying to make a new polio vaccine that doesn't depend on the presence of live virus or even virus sequences, i.e., it's a VLP vaccine. So the overall aspirations and justification for this project was pretty straightforward. Is that a VLP, the advantages over other poliovirus vaccines is that there's no virus involved in its manufacture. No containment, therefore, required for the manufacturer, no high-level containment. And there's no risk of seeding outbreaks. The objective was to produce material at low cost, which, and of course, doesn't need any inactivation or high containment facilities, as I said. And hopefully something that can be produced rapidly and in sufficient use to be competitive. The other point is that we were trying to aim for a technology that could be readily widespread and transferred to other countries other than the richest countries, i.e. inexpensive plant. Now, in principle, it's childishly simple. It's just make a VLP vaccine that replaces the IPV vaccine, but doesn't obviously contain any nucleic acid. And we've known for many years that the poliovirus and other enteroviruses produces empty capsids, which are antigenically indistinguishable from the virus and therefore potentially effective as vaccines. But the drawback of that is that they are inherently much less stable than the virion and convert from the effective D-antigenic conformation to an ineffective C-antigenic conformation readily, and probably are insufficiently stable and robust with down manufacturing processes. So the first part of our consortium's activities were to identify ways of introducing mutations into the capsid sequences that will give a more stable, antigenically stable capsid particle without disrupting any of the antigenic sites. And so, let's say the first part of our consortium's work was to address this issue, and we tried several routes, but the best one evolved was, in fact, developed by Andy McAdam, which was simply to take a temperature-sensitive strain, grow it at a non-permissive temperature, and identify the mutations that arose, which enabled it to survive that. And a whole number of mutations were therefore identified in individual viruses that broke through this restriction. Those individual mutations were then put in various combinations artificially into the capsid sequences, and the consequences on the stability of the particles measured. Having gone through this procedure, the next step was to try and produce these VLPs by recombinant expression systems, and we've used a whole variety of mammalian cells, yeast, insect cells, plants, and as you'll probably hear next, in fact, in cell-free systems. So this demonstrates the inherent problem with empty capsids. On the left, you see the antigenic activity or reactivity of virus particles as you progressively heat them, as you can see with two monoclonal antibodies which recognize either the antigens diagnostic of the D-antigenic form, the virus form, or C-antigenic, the ineffective empty capsid form. And you can see there's a switchover temperature at about 45 for the virus and much, much lower for empty capsids. And it's to push that switchover temperature up higher that the first part of our activities were directed against. And this summarizes the number of mutations that have been introduced into the structural protein structures of PP1, PP2, and PP3 to give these mutant complexes. The particles produced with these mutations in them, we've solved all of those by cryo-EM technology, and they are essentially indistinguishable from the virion in terms of their external appearance and structure. And as you can see here that the green objects highlighted are the antigenic sites, and the red are some of the stabilizing mutations, and there's no interference between the two. So although the particles are more stable, they are antigenically unaltered. And also the VP1 lipid stabilizing factor is present in nearly all of our expressed particles, apart from those produced in plants. Rather surprisingly, particles produced in plants seem to have an empty pocket. Here we see the baculovirus expressed empty particle in the middle there, with the sphingosine stabilizing lipid naturally present in the pocket. And to the right there's the that sphingosine that's produced in yeast-expressed VLPs, and also you can replace it with a stabilizing drug, GPP3, as you can see on the extreme right there. And those, so it's possible to further stabilize the particles by using an appropriate pocket-binding drug. Another way to further stabilize is to occupy a pocket, the TSS, glutathione binding pocket, on the surface of the particle, which is the structure which is shown here. And that, as I say, further stabilizes the thermostability of the empty particles, VLPs. So having identified ways of making more sufficiently, we believe, stable particles, the next trick was how to produce them. And all of our VLPs are synthesized by basically the same construct, which is to have the P1 structural protein precursor and the poliovirus PV3CD protease expressed on separate from separate parts of the construct, either in plants or in piscia, as we've shown here. Same overall method is used for baculovirus. So basically, it's a two-component expression system with the structural proteins and the protease separated. We found that we've been concentrating mostly on yeast recently, and we find that we can get reasonable amounts of material by expressing in a laboratory scale flask culture, but some experiments using a more controlled bioreactor fermentation gives significant increases in yield of material, and we believe now that the yields of particles are at least similar to the yields that are obtained in conventional current IPV production for polio vaccines. Purification, till now we've mostly used lab scale, ultracentrifugation based purification methodologies, but which are not really suitable for further development towards industrial production. And so we're exploring the column-based purification methods. And recently we found that using a glutathione affinity column is remarkably effective for at least two of the serotypes of the virus. And what this shows here is the wash and elution of particles from very crude extract of the materials and just simply pass through a column. So it looks as though we can get a very simple purification in a one-step process. We're also obviously concerned by the presence of nucleic acids within these particles. We had no reason to believe that there should be nucleic acids there, but we've checked it out as far as we can. And this simply shows that using a fluorescent dye-based methodology, the pastry assay, that in brown there you can see as you heat virus particles, you get clear increase in fluorescence due to exposure to the nucleic acid. And then the lines below that all of which are on top of each other, shows that with the VLPs, you see no signal detected for the presence of a nucleic acid. So as far as we can determine, these particles are empty. There's no nucleic acid within them. So the key issue, of course, is the immunogenicity of them. And so we've been exploring two models. The transgenic mouse model has been referred to earlier. And using this model, we find that our particles introduce neutralizing antibody titers that are at least as good as that induced by IPV. And we have very, very good protection upon challenge. And there's similar results with each of the three serotypes. Model 2 is the perhaps more important one because it's the batch release assay, which is the immune response in rats. And here we found a bit of a disappointment that without adjuvant, the VLPs were less immunogenic, and the neutralizing antibody titers that we induced were variable and mostly less than IPV. However, use an adjuvant and a very simple, highly well-used adjuvant, aluminium hydroxide, we get much improved responses. This just demonstrates model 1, the transgenic mouse, where we can see that the VLPs are, in fact, significantly better in this case than IPV in terms of both the induction of neutralizing antibodies and survival on challenge. And this shows the immunogenicity in rats, where the purple line shows the non-adjuvanted yeast trivalent particles, where we get pretty less response to all three serotypes than we do with IPV. However, with the simple addition of aluminium hydroxide, that's converted to a much, much better response, in fact, higher than we see with IPV. So that briefly summarizes what we've achieved so far in this consortium. Anyway, all the very best, Kostya, and may your retirement be long and fruitful.
We started looking at the ability to actually study the assembly of VLPs in the cell-free replication system, which is really HeLa extracts that were originally basically the same that Ellie Ehrenfeld developed many years ago for doing in vitro translation reactions, but they also will replicate the input RNA if you look for that and optimize the reaction for that. So when we started to think about this, we wanted to optimize the possibility of actually making VLPs, and you can see that we could actually do that in our cell-free reactions. And basically, these are what we were thinking of as the requirements: construction of a poliovirus RNA expression vector that would meet the following requirements. That's we would have to synthesize the P1 capsid precursor protein, so we'd have processing would release the capsid proteins. We would need both proteases, 3CD Pro and 2A Pro, for release of the P1 and then the subsequent processing of P1 to form the capsid proteins. And so the 2A Pro, in other words, that will actually allow you to put everything in one piece of RNA, because it will separate the P1 precursor protein from, you have to have 3CD in order to process that. But so that's one reason you need the 2A Pro activity to release the P1 from the rest of the protein coding sequence. And then in our studies with the cell-free system, we found out that the 2A Pro activity itself increases translation and RNA replication in the cell-free reactions. And we aren't quite sure. It's actually something that's required. It requires the catalytic activity of 2A Pro. If you don't have the catalytic activity, you don't see that. And that's above and beyond, and then these, in the cell-free reactions, you don't have to worry about inhibiting translation like you do in the cell, because there's the cellular messenger RNA has already been degraded. In addition, the 2A pro activity is required to stabilize the poliovirus RNA, and we had learned that as well from our studies in the cell-free reaction. And this is something that's very specific for poliovirus RNA, and we never figured out exactly what it's doing in that regard, but we first thought it was just digesting our RNases, but in fact, it's specific for polio. We tried it with some other viral RNAs, and it had no similar effect. And then finally, we wanted to inhibit viral RNA replication and packaging, because we didn't want to get any of this RNA actually packaged into any VLPs that were made in the reaction. And we didn't want the RNA to replicate. If it doesn't replicate, it's not going to package. But in addition, we didn't want it to replicate because we didn't want it to undergo recombination. If it was ever used in some, for any purpose outside of the cell-free system. Okay, so those are the basic requirements that we were thinking of. So the vector that we came up with is shown here. And basically the idea was we just took a transcript of poliovirus RNA. And the idea was we need the capsid precursor protein P1. We needed 2A protease, and we needed 3CD protease. So we deleted, what we did is we made an in-frame deletion between 2A and the beginning of 3CD. And basically that left those proteins intact, 2A and 3CD, and took everything else out in between. And that made it, basically we removed some very key proteins that are absolutely required for RNA replication. So that ensured that it wouldn't replicate, but just to make sure, we also added a mutation in the 3' NTR, which we had shown in our replication studies, which would also block replication. So the idea was then we could just translate this RNA in the cell-free system, and it should then make the appropriate proteins, and we show us, I've got a gel here where you can see that we do get the proteins that you would predict. We have the 3CD, we have BP0, and then we have BP1 and BP3, and those are the 3 proteins that go into making empty capsids. And then we have the 2A protease, and then we have some 3D prime, which is another protein that gets is made from cleavage. In terms of, it's not thought to be required for replication, but you do see that normal with translation of poliovirus RNA. So we were getting everything just exactly what we predicted from this construct. And then we looked in several ways, and here's an electron micrograph, and we could see the VLPs were present. This was a sucrose gradient, partially purified VLPs. So then the question was, what would be the strategy for making stabilized VLPs? The problem comes up in terms of, we knew that for a long, long time that you get empty capsids in infected cells, poliovirus infected cells, but those empty capsids aren't stable at physiological temperatures. So that really wouldn't be much use in terms of making those empty capsids for vaccine purposes. And so basically, you can get, in other words, you would get your P1 protein, you have 3CD processing, so you have BP0, 3, and 1, just like I showed, that can self-assemble into using the normal pathways. You have your P1 gets processed, you have protomers and pentamers. The pentamers can then assemble into empty capsids. And these other steps would not occur because we're not any new RNA, so we don't have any genome replication. So you don't go through forming the provirion or the virions in this case. The problem here is this empty capsid is not stable at 37 degrees or above, and so it falls apart very rapidly, and you lose all of the antigenic potential there. So the idea was, just like Dave Rowlands just talked about, and they've really had great success in making a lot of mutants that help stabilize the empty capsids. And so that was the general idea. And at the time when we were doing this and basically working independently from Dave and the UK consortium, we wanted to come up with ideas about what could we use for a mutation that would stabilize the capsid without preventing it from assembling or change its structure. So being an old virologist, even at that time, I was familiar. One thing, you've read a lot of papers. So I remember Vince Racaniello had worked in some of his work. This mutant played a starring role in one of the papers. So it was previously identified by Elizabeth Colston and Vince, and it was published. And it was a soluble receptor resistant mutant. And these mutants were resistant to neutralization by a soluble form of the poliovirus receptor. Normally, if you had a lot of soluble receptor, and Vince can correct me on this, but you have a lot of soluble receptor, that's going to basically neutralize the virus because it fills up all the binding sites. But if you get, in other words, you would prevent labeled wild-type virus from binding. But if you could find mutants, that would suggest that the receptor, the soluble receptor, wasn't binding. So basically, in the discussion of that paper, which I read with interest at this, reread it again, it was suggested that the mutants like BP3Q178L may actually strengthen interactions but at the promoter interface and thereby reduce structural alterations in the virus capsid. And we used our vector and constructed that mutation, that 1 point mutation, into our protein expression vector. So we saw similar amounts of VLPs that were formed with the mutant and the wild type and set them in exactly the same place. And then we looked at the antigenicity, and this was using a monoclonal antibody that was obtained from commercial sources and showed that they both had same levels of inantigenic activity. And we were supported all along the way with funds from the WHO and the Polio Global Eradication Initiative. Thank you.